Alright, exam 4, essay question number 2. Let's go ahead and read it and get into this. It's pretty heavy. Well, if you're like me, it is. Alright, explain tubuloglomerular feedback, explain the renin, angiotensin, aldosterone system, describe what happens at the cellular level with ADH and aldosterone. In part 2 of this essay, explain how the loop of Henle is used to establish and maintain the renal medullary concentration gradient. Explain how this gradient and ADH can work together to excrete urine of different concentrations. Okay, first. So, our tubuloglomerular feedback system, this is basically how we can regulate the rate of glomerular filtration, and that's going to make sure that it stays stable, because we want things to stay stable. There we go. Okay. Camera was bouncing around. That was weird. Maybe it's just me. Anyways, so let's look first at our apparatus. It's called the juxtaglomerular apparatus, and that is made up of the macula densa and the cells supporting the macula densa cells. So these cells will detect our salt levels, our sodium and our chloride levels inside of the distal tubule. Let me go back. This apparatus is located in between our ascending and descending distal tubes. So if there's a higher salt concentration next to this distal tubule, that's going to indicate a higher filtration rate. And then these cells will signal to our brain using nitric oxide or let me go ahead and check. Jeez. I have it pulled up here, the names. I just wanted the names of the chemical signals. It will use adenosine. There it is. Okay. So yeah, nitric oxide and adenosine, and using those two things, it'll affect our filtration rates. So if our cells signal, if our macula densa cells signal that there's a lot of filtration going on, then they will use this adenosine to shut that down, to get those arteries around our filtration to constrict, and so filtration will be stopped. Likewise, if our salt levels are low when they detect that, they're going to use nitric oxide to signal to the brain to vasodilate, and that will increase blood flow, and that will increase our filtration. All right, so that's it for the, gosh, I hate this name, the juxtaglomerular apparatus and our tubuloglomerular feedback system. More salt, less salt, constrict or dilate, filter more, filter less. All right, let's get into the renin, angiotensin, aldosterone system. I'm going to call it the RAAS, much simpler that way. And this is very important for our blood pressure regulation and our fluid balance. So when our blood pressure will drop, our kidneys are going to release renin, and renin will convert into angiotensinogen. There we go. And that is a protein that's produced in the liver. And so the angiotensinogen, gosh, that'll be converted into angiotensin 1. And our angiotensin converting enzyme will then convert our angiotensin 1 into angiotensin 2. And that enzyme is found normally in the lungs. So once it's converted into angiotensin 2, it will have different effects on our bodies. First off, it will constrict our blood vessels, to increase our blood pressure. And that'll also stimulate our adrenal cortex to release aldosterone. So once aldosterone is out there, that's going to promote the kidneys to reabsorb more sodium and water. Go watch video 1 for a little explanation there. And that'll increase our blood volume and pressure. So if there was that pressure drop, we can account for it by having more sodium and water pulled from our urine stream or our filtration system in the kidneys to give us more volume. All right, ADH and aldosterone. So ADH, or our antidiuretic hormone, that's going to be released from our pituitary gland, and that's going to bind to our receptors on the cells of the collecting ducts, and that's going to cause our aquaporin channels to insert back into the membrane. And this will increase our water reabsorption back into the bloodstream, and that will concentrate the urine even more. Vice versa with aldosterone. Aldosterone is going to use the distal tubules and the collecting ducts, and that will increase the reabsorption of sodium and excretion of potassium. So sodium is going to be reabsorbed back into the body, and potassium is going to be kicked out into our urine system. By reabsorbing our sodium, our water is going to be pulled as well via osmosis, and that will increase our blood volume and pressure. All right, I think that covered everything in part 1, ADH and aldosterone. Okay. Explain how the loop of Henle is used to establish and maintain the renal medullary concentration gradient. Explain how this gradient and ADH can work together to excrete urine of different concentrations. Super fun. Super exciting. All right. So the loop of Henle, its descending limb is, as we know, and its ascending limb is permeable to our salts and our ions. So as the filtrate descends, the water is going to exit into the medulla, and that's going to concentrate our filtrate. Filtrate. Wow. And then as it's ascending, in the ascending limb of the loop, the salts and our ions are going to be pumped out into the medulla, making the filtrate hypertonic. Making the medulla hypertonic, excuse me. All right, the countercurrent multiplication mechanism is going to make a gradient where the medulla's interstitial fluid will be more concentrated, deeper down it goes, further towards our bladder. ADH is going to use this gradient, and it's going to regulate the urine concentration. So when ADH levels are high, the collecting ducts will become more permeable to water, and that will move out into the concentrated medulla, making more concentrated urine. When ADH is low, the ducts will remain impermeable, and that will dilute our urine, just because it's not being concentrated, obviously. I think I covered all of that with the ADH system. Work together, create urine of different concentrations. I think I did. All right, back here for question 3.